Hi everybody, welcome back again. I'm once again at my school computer just because I want to get this lecture out there. So I apologize for the microphone quality, but it's more important to me that we get it out there in a timely manner. So today's lecture is going to be on DNA replication and the structure of DNA. Both of these are basically foundational for the rest of the unit. Sometimes it's going to seem a little sporadic how we're talking about things, but once we get to later parts in the unit, it's going to make sense why we're talking about them now. So let's go ahead and start. So you should already know, based off of the history lesson and last year, that Watson and Crick determined the base pairing rules. And so you should know that there are 4 nucleotides in DNA: adenine, guanine, thymine, and cytosine. In RNA, instead of thymine, we see uracil. And you should understand that they exhibit specific base pairing rules. A pairs with T, G pairs with C, and in RNA, A will pair with U. Now, the main part here that you need to understand is that this is what varies from DNA strand to DNA strand. So the sugar phosphate backbone, which are the other 2 parts of the nucleotide, are actually the same in all DNA molecules. That is not going to be different, but the actual base is what varies from person to person or organism to organism. Okay, now this should be somewhat new. We're going to actually separate these 4 bases into 2 categories. They are either going to be pyrimidines, which are the single ring nucleotides, which are your T, C's, and U's, or they are going to be purines, which is the double ring nucleotide, which is A and G. Now, I wish there was a really good way to memorize this. Some people have come up with clever mnemonics, but frankly, it's only 5 different bases that we have to worry about here: cytosine, thymine, uracil, adenine, and guanine. So you might just have to commit that to memory, which ones are pyrimidines and which ones are purines. But the way that I remember the structure themselves is I say smaller word, bigger base. So purine is a smaller word. These are bigger bases. A and G are bigger bases. And then you can say bigger word, small base, and you can use that for the other side as well. That's how I would do it. It's up to you. Ultimately, it's not that much to memorize, but it can be a little tricky. Let's keep going. So, yes, Watson and Crick, they said that they figured out that thymine and adenine pair together, but it was actually based off of the work of Chargaff. So Chargaff figured out in double-stranded nucleic acids that the percent of A and T is roughly the same. And the same is true for G and C, that they are roughly the same. And this was true for all species that we now know. And so this laid the foundation for, of course, what Watson and Crick eventually said was their base pairing rules. Unfortunately for Chargaff, he didn't really get a lot of the credit, and he spent most of his life reminding others that he was the one who figured it out, and then it was stolen, and that he should receive credit. Pretty rough life for Chargaff, but nonetheless, it was a very important discovery. Okay. So typically what you're going to see with these types of questions are basically just simple math questions. And so if I were to ask you the percentage of adenine based off of the finding of a percentage of thymine, you should be able to tell me that it's also 21%. So in this case, you find a new type of bacteria and sequence the genome, and you find the percentage of thymine is 21%. So what's the percentage of adenine? Well, because of Chargaff and Watson and Crick base pairing, you should understand that thymine and adenine are roughly the same. So 21% thymine means 21% adenine. Now, sometimes you're going to get slightly more tricky questions. This is not too difficult, but it's a little bit harder. So a geneticist sequences the genome of a lion and finds the percent of guanine is 34%. What's the percentage of adenine? This is a little bit trickier because guanine and adenine don't necessarily equal each other in a DNA strand, and that's going to be the case here. So how do you do it? Well, you say, okay, guanine is 34%. That means that cytosine is also 34%. Together, that's 68. You subtract that from 100, and you get 32% for the AT content of this DNA strand. Well, if A and T are the same, you can just divide the 32% divided by 2, and you'll get 16%. That means A is 16% and also T. That's the long way to do it. That's the way that I typically do it. But the rough and ready way is you can just take this number, 34%, and subtract it from 50, and that'll get you it as well. And you can work through that. If you don't believe me, you can work with it with all sorts of numbers, and then you can convince yourself. Okay, now let's talk about actually the physical structure of DNA. So we know the base pairing rules, but let's talk about the actual structure. So you should know already that it is a double helix. And thanks to Rosalind Franklin and the work from Watson and Crick, we discovered that. And you should also understand that these nucleotides, this phosphate sugar backbone, is connected covalently. Covalent bonds are significantly stronger than hydrogen bonds, and hydrogen bonds are what hold the bases together. So maybe you can think about why that's actually advantageous, that the stuff holding the helix together is hydrogen bonds, whereas the backbone is sugar phosphate. Well, considering that the sugar phosphate is the same for all organisms, it makes sense that those should be covalently bonded, very strong bonds. And when we get into DNA replication, it's going to make quite a bit of sense why these need to be hydrogen bonds and a little bit weaker. I want you to keep that in mind as you're working through the whole unit, why this actually makes sense evolutionarily and chemically. Okay, now, if you remember all the way back from Unit 1, I really harped on this because remember, my background is in lab science, so I worked a lot in a lab in undergrad and a little bit in my grad school. So I care a lot about the practical implications of our findings, and this is a really important practical fact that you should know. So how many hydrogens are there between guanine and cytosine? There are 3, and there are only 2 between A and T. Well, the fact that there is a difference actually has a lot of practical implications in a lab, especially when we're dealing with something like PCR. So when I have a DNA strand, if it has way more guanine and cytosines, maybe like 64% guanine cytosines, that's heavy in guanine cytosine, which means there are actually more hydrogen bonds in that DNA strand than maybe a 50-50 split. Now, why does that matter? Well, hydrogen bonds, while they are weaker than covalent bonds, they are still going to take energy to separate. And so when I do something like PCR, I'm going to have to add more energy, aka heat, to actually separate those 2 strands if it's really high in GC content. Whereas if it's low in GC content, I can actually have a much lower temperature to denature that DNA strand. That's why it matters. It's really, really important in a lab. Okay, that's what I was saying here. The hydrogen bonds between the bases, well, there are 3 between G and C, 2 between A and T, and this has some pretty important implications. If you need to ask me questions, please feel free to come after school and I can explain this again. Okay, so looking at this question that I gave you in class, which DNA molecule would require less energy to denature or unwind? How I approach these questions is, one, I try to reword it, and I say, okay, what is this actually asking me? Well, denature means to split open in terms of DNA, and so which one requires less energy? I'm looking for the one with the lowest GC content. So what I'm going to do is I'm going to change all of these to GC. So this first one, 32% AT, that means that 68% are GC. B is 32% GC, no work I have to do there. C is 30% GC because it's going to add up to 100. And then D is 70% GC. So the one that's going to be the lowest GC is actually C. It is really easy to fall for a trick here and just pick B because you see a low number with GC. But if you actually do the math, GC content for C is 30%, and that's lower, so it's going to take less energy to denature. Okay, now we're going to go ahead and get into some of the tricky language that is used when we're talking about DNA replication. And the first word we're going to look at is antiparallel. So antiparallel means that the 2 strands, because DNA is double-stranded, they are going to run opposite to each other, and they are flipped in orientation. Now, instead of using terms like right and left or up and down, we actually use the verbiage 5' and 3'. Now, where do we get these numbers from? It's not just because we really like these numbers. It actually has to do with the naming convention of the carbons. So if we look at the sugar here, this is my deoxyribose sugar. We start here with the 1', and you're just going to have to believe me that some organic chemists decided that that's where we're going to start. So this is the 1' carbon. 2 going clockwise, 3, 4 is this point, and then 5 is the methyl group connected to that point. So this 5' carbon then is connected to the phosphate. Now if we go all the way to the other side of the DNA molecule, we still start here with the 1. The 1 is connected to the nitrogenous base. 2, 3, and this side is connected to the hydroxyl. So if we are reading this DNA strand from one end to the other, the furthest end on one side is the phosphate group, and that's connected to the 5' carbon. If we go to the other end of the molecule, the furthest group on this molecule is the hydroxyl group, and that's connected to the 3' carbon. So that is why we call it 5' to 3'. It's because the last groups that are connected to carbon, on one end is the 5' phosphate, the other end is the 3' hydroxyl. That's why we have that. Now if we look at the other side, we'll notice that one end is still the hydroxyl and is connected to the 3'. The other side is going to be the 5' phosphate. These are always going to run opposite to each other. So if you have 5' to 3' on one side, then it'll be 3' to 5' on the other side. And that's what you'll see in your modeling thing that we did in class. It's pretty helpful. Okay, now let's go ahead and look at the 3-dimensional structure of DNA. Whereas before we were looking at the 2-dimensional structure, just sort of a drawing, a schematic of what DNA is like. I want you to understand that DNA is actually a very large molecule. It has a 3-dimensional structure, and because of that, some interesting features can arise. So when the backbones are close together, we call that the minor groove. This is the sugar phosphate backbone. It is relatively close together. Whereas when they are further apart, we call that the major groove. Now, I will never show you a DNA molecule and expect you to tell me which is the minor and which is the major groove. However, I do want you to understand that when we have 3D structure, we get new functions that arise, right? And so this is really important because we have that adage that we say over and over and over again, that structure dictates function. Well, DNA interacts with lots of different molecules. It interacts with enzymes, it'll interact with small groups like acetyl groups and methyl groups, which we'll see during epigenetics. But it interacts with lots of molecules, and these molecules are specific, just like we've seen in everything else, that these molecules are specific. So some might interact at the minor groove, and some might interact at the major groove. Whether or not you can distinguish them is unimportant at this level, but I want you to understand there is a 3D structure, and then that is going to end up dictating at least some of how the molecule behaves and interacts with other molecules. Now, I also want you to understand that we're going to be talking a lot about DNA for basically the full week of classes, right? We're talking about mostly DNA, but I do want you to understand that some things use RNA as a genetic material. And the thing that uses it is the thing that is usually the exception in biology, which is viruses. Viruses can use RNA as a genetic material. They can use DNA as a genetic material as well. There are lots of different types of viruses, and you only will know if you've really tested the genome what type of genome it is. It could be double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA, and the actual orientation of those molecules matters as well. So viruses are pretty fun, they're pretty weird, but I want you to understand that they can be used as genetic material. But we also need to understand that for the most part, we are focusing on eukaryotic organisms in AP Bio. That does not mean we won't talk about prokaryotic organisms. We will at certain parts of this unit, but for the most part, we are focusing a little bit more on eukaryotes. But there are some things I need you to know now so that when we talk about prokaryotic replication and transcription translation, it's not a shock later. So the first thing that I want to remind you of is circular versus linear DNA. Prokaryotes typically have circular chromosomes, whereas eukaryotes typically have linear chromosomes. And that's going to become really important when we talk about a feature of eukaryotic replication in a second. The other piece here is plasmids. Prokaryotes often contain plasmids. That's just going to be extra chromosomal DNA that typically prokaryotes pick up from their environment and they can work with it. Eukaryotic organisms can sometimes do this as well. For example, yeasts can take up some plasmids, but generally speaking, and yes, this is massive generalization, it is more associated with prokaryotes. Okay, now because DNA and RNA can both be considered genetic material. I just want to remind you of some of the differences. Now, I hate this schematic because it looks like they're saying this is DNA and this is RNA, and no, if you look at the bottom, they're saying this one is RNA and this is DNA, but just graphic designed a little poorly. But there are some features you need to know are the differences, right? The bases are different in DNA. Adenine, guanine, and cytosine are the same, but in DNA we have thymine, in uracil we have, and RNA we have uracil, my bad. The actual physical structures are different. We have single-stranded RNA versus double-stranded DNA, and the sugars are different. That is a huge, huge difference that becomes important later. But the sugar in RNA is ribose, and DNA is deoxyribose. Very important differences. Yes, you need to know it, but it should be reviewed, so don't stress too much. Okay, now let's go ahead and start on the tricky stuff. So hopefully most of the DNA structure was reviewed, and now we're going to get into DNA replication. Now let's go ahead and break down the word DNA replication. Well, to replicate is to copy, to make a copy. It's the same root as replica, so you're making a replica. And I want you to remember that our cells do it during S phase. So whether or not you need this reminded is up to you, but generally speaking, you guys should know this, and you should also know that it occurs during the S phase because we talked a lot about it in Unit 4. Okay, now the way that DNA replication proceeds is through a semi-conservative fashion, this middle column here. But we didn't always know that. Instead, in 1958, the Meselson-Stahl experiment showed it. So the competing theories were conservative versus semi-conservative or dispersive. And in the conservative model, you would have one totally brand new DNA strand using the original parent strand as a template on both sides. And how this worked is a little bizarre. Semi-conservative, each of these strands ended up working as a template for the daughter strands that we see here, and they end up, each molecule was half old, half new. And then the dispersive, very bizarre. I don't know the mechanism that was predicted here very well, but it basically looks like a tie-dye of the new and the old. Okay, so how did they actually distinguish between the three? Well, let's look at it. Now, generally speaking, what they did is they grew E. coli in nitrogen 15. This is a heavy nitrogen, and so all of the DNA in that E. coli incorporated heavy nitrogen. Then they removed those E. coli cells and allowed them to grow with light nitrogen, N14. So they started with the parental strand like we saw on 2 slides back, and if it was conservative, what they should have seen was a band that was entirely old and one band that was entirely new. So that would mean that none of the N15 would be incorporated with any of the N14. This is the light nitrogen. Now, after the first replication, they didn't see that. They didn't see a band for the N15 and the N14. Instead, they saw a band for a hybrid. So 50% of the DNA molecule was N15 and 50% was N14. So they only saw one band. So after one round of replication, they actually already knew it couldn't have been the conservative model. But then we're between the semi-conservative and the dispersive because both of these would be hybrids of the old and the new strands. So that's what we see here. So they need to do a second round of replication. We don't really worry about conservative anymore. It doesn't matter because we already know that's not the case. So just looking at semi-conservative and dispersive, well, if we do another round of replication, well, this yellow strand, which is the N14, would be incorporated with another N14 because that's what's available. And so you'll have one strand that is totally N14. And you should see a band that is totally N14. And then you should see another one where you have the original parent strand all the way from back here, and that would also be with N14. So you should see 2 bands, one that is N14 because that's what they're being grown in, and that would be the daughter strand from replication 1, and you should have your parent strand from generation 0 with a new strand from generation 2. And so that would be the N15, N14. And that's what they saw. They saw this, and so they were pretty confident. And then if you could expect what dispersive would look like, well, the dispersive should still be 50-50. Now the bands might migrate up because you're incorporating more N14, so it would be technically a little lighter than the previous generation, but you should still only see one band. So that is how they proved it. It's a really elegant experiment. It's a very well cited experiment because it's so good at proving what they set out to prove. Okay, so hopefully that was a good enough explanation for you to understand what semi-conservative is. The correct answer here is going to be B. Each new DNA molecule consists of one old strand and one newly synthesized strand. So it's not a totally old double strand, another totally new double strand. Instead, it is going to be one that's half old, one that's half new. Okay. Let's go ahead and start talking about how this actually works. So if we have a semi-conservative model, that would mean that each of the original strands are going to act somewhat like templates for a new strand, and then eventually it'll pinch off and you'll have a partially old and partially new strand. So how does this happen? Well, I just want you to think generally. What has to happen here? I need to have something that splits open my strand so that we can access those parent strands. I need to have something that is going to add new bases and new nucleotides. I need something to make sure that it doesn't close back up. I know that I'm going to need something that's going to glue it all back together. All of these things are generally, and hopefully it just sort of makes sense logically that would happen, but we're going to talk about it a little more specifically. Before we do that, we need to get some vocabulary out of the way. The first is origin of replication. As the name suggests, origin refers to starting point of replication. So this is a starting point of replication. So this is where replication begins. Now, when these two strands separate, we are going to generate the replication fork, very similar to a fork in the road. Now, the fun part here is that you can actually have lots of origins of replication because it would be very, very, very slow considering how large our genome is. Our genome is very large in comparison to something like a prokaryote. We need to have lots of origins of replications and lots of things opening these up and adding in new nucleotides so that we can be very efficient. And so that's what we have, and all of these origins of replications make a replication bubble. So these bubbles, they're all throughout our whole chromosome, and it makes it so that replication happens much more quickly. Now, this does not happen in eukaryotes. If you have a circular chromosome, it's typically copied with one origin of replication because they're much smaller. They don't need to have several origins of replication. Okay, now I keep using this term things, and that's intentional because I want to start planting a seed for this. What macromolecule allows for basically all processes in a cell? Well, hopefully your mind went to protein and then more specifically to enzymes. And so we're going to be talking about the enzymes that are involved in DNA replication, and I want you to remember that they typically end in ase, and you're going to see that is the case for these enzymes as well. So don't forget, enzymes are so important and they're not going away. Okay, so let's talk about these enzymes. Now, I like to talk about DNA replication in two stages. This is me. This is a my thing. This is a Ms. Geber thing. This is not something you're going to see in textbooks and something you're going to see in other videos. This is just the way I like to break it up. So I like to think of it as the setup phase and the extension phase. And the setup phase is basically everything that has to happen to the DNA before we can start adding any new nucleotides. And that's quite a bit of work. You can see there's lots of enzymes here. Whereas the extension phase is the time where we're actually making that new DNA strand. So that's how I like to break it up. We're going to go ahead and move forward. Let's start talking with a very popular enzyme. Lots of students love this enzyme, helicase. So helicase is the first enzyme in the setup phase that is going to unwind the DNA. So remember that DNA is double-stranded we need to separate those two strands. Those two strands are actually held together by the bases. The bases are connected to each other through hydrogen bonds. So helicase is going to be the enzyme that breaks these hydrogen bonds to actually split the DNA strand in half. This is a very important enzyme. You can imagine without this, we can't do any DNA replication. So it's an easy enzyme to remember. Helicase, same root as helix. It unzips the double helix by breaking hydrogen bonds. Pretty easy to start. Let's keep going. Now I want you to imagine that you have two strings that are taped to your table, and they have been spun up really, 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 really tightly. Spun, 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 spun. Two strings. Then I want you to think, okay, if I pull these two strings apart really quickly, what's going to happen? Well, one, I could break the strings. But two, and the more concerning one, is that these strings, because they're all twisted together, actually might twist on top of themselves. Much like if you twist your hair really, really tightly, you bring it close to your head, it'll coil on top of itself. That's called supercoiling. And it means that we are not going to be able to replicate the DNA. If it's supercoiled, now these bases are inaccessible. Even with helicase there, you can think of helicase, the thing that is splitting the two strings apart. That means that we need to have another enzyme there preventing supercoiling. And that is what topoisomerase does. Topoisomerase is one of my favorite enzymes. It is a really cool mechanism. It kind of reverses the twist that is happening in the DNA to prevent that supercoiling from happening. So these two work together to ensure that the DNA is being properly unwound. Now, you might be already noticing something on this model here to the right, these little purple things, and that's what we're going to talk about next. So these purple things are called single-stranded binding proteins. Sometimes you see it shortened to SSB, sometimes you see it shortened to SSBPS, so single-stranded binding proteins, but if you don't see it shortened, that's kind of the best because the name gives it away. Single-stranded binding proteins. So once helicase unwinds the DNA, well, DNA was double-stranded, but when helicase splits it, now these are effectively single-stranded. So these proteins bind the single-stranded DNA, and they do two really important jobs. The first one is that they prevent the DNA strand from coming back together. Remember, hydrogen bonds, they are weaker than covalent bonds, but they are still quite strong in terms of intermolecular forces. So these DNA strands are going to want to come back together. They also do something else that's really important. They prevent these single strands from being cut from something called nucleases. Well, you might notice the last three letters here, or last four letters, ACEs or ACE. That's probably an enzyme, and these are specifically enzymes that cut nucleic acids. There are lots of nucleases, and they are all specific to lots of different types of nucleic acids, meaning single-stranded DNA, double-stranded DNA, single-stranded RNA, double-stranded RNA. But understand that they have some practical uses. For example, if you have single-stranded DNA, that could be viral DNA, and that's foreign, and so you want to have nucleases that can cut those. Problem is during replication is we temporarily have the single-stranded DNA, and we need to make sure it doesn't get cut. So that's the other role of single-stranded binding proteins. They prevent it from coming back together, the two strands from coming back together, but it also prevents damage from nucleases. So far we have three different important proteins here that are important to the setup phase. Okay, now here is another fun enzyme. This is primase. Primase is an enzyme that places an RNA primer for a DNA polymerase to bind with. This is not a typo on the screen. Yes, it places an RNA primer. And this is in part because primase is an RNA polymerase. So what does that mean? It actually only recognizes RNA nucleotides. And RNA and DNA are quite similar with the exception of the sugar in both of those nucleotides. So RNA uses a ribose, DNA uses a deoxyribose, and the difference is really just one hydroxyl. And so this is really important. So primase is an RNA polymerase. You don't necessarily need to know that, but I think it's important to understand why we use an RNA primer. DNA polymerases aren't capable of initiating DNA replication. So we have to use what we have in our body, which is an RNA polymerase, aka primase. Now you might be asking, okay, what is an RNA primer? Well, an RNA primer is just a few RNA nucleotides. It's just a couple of RNA nucleotides that are there, and then you can use that to build off. DNA polymerase can recognize that and then build off to make the rest of the DNA molecule. This is very important. You do need to know this. This is something I got wrong when I was taking AP Bio, and it still haunts me to this day. I said it was a DNA primer. It is an RNA primer, even for DNA replication. Okay. So those are the setup enzymes. Now let's start talking about the extension enzymes. So the first one is DNA polymerase 3. And again, I'm sorry, it just happens that the numbers work out that way. But DNA polymerase 3 is the first one, is the major player. It is the one that adds most of the nucleotides complementary to the parent strand. Now, here's the thing about DNA polymerase. DNA polymerase only works in the 5' to 3' direction. Now, recall all the way back earlier in this video, in any DNA strand, we have one side that's 5' to 3' and the other side that is 3' to 5'. Uh-oh, how's this work then? Well, I want you to think about the easy side first. So the parent strand that is in 3' to 5' will generate a daughter strand that is 5' to 3'. So DNA polymerase can very easily sit on top of the 3' to 5' parent strand and synthesize a daughter strand in 5' to 3' direction. This is the easy side. But DNA polymerase 3 works on both strands. It works on both the 3' to 5' parental strand and the 5' to 3' parental strand. So how does this work? Well, it's going to generate two strands for replication, the leading and the lagging strand. So let's look at the leading strand. That's the easy one. DNA polymerase sits on top of the 3' to 5', and then it can happily work in the 5' to 3' direction that it wants to work in. The lagging strand's a little difficult. So instead of working this way, so looking at my pointer instead of working this way, which is 3' to 5', DNA polymerase can't do that. So what it has to do is it has to wait for helicase to open up just a little bit more and then work backwards. Helicase opens a little bit more, works backwards. That way it is staying true to its 5' to 3' direction, but it always ends up making these kind of clunky Okazaki fragments. So if you just follow my pointer here, I want you to think my pointer is, well, it's going to change roles. It's going to be both helicase and polymerase at certain times. So helicase opens up, DNA polymerase can bind, it'll work backwards so it can work in the 5' to 3' direction. Helicase opens up, polymerase binds here, it works backwards to be in the 5' to 3' direction. Helicase opens up, it binds again, DNA polymerase binds again, works backwards to work in the 5' to 3' direction. These fragments are called Okazaki fragments, and they form the lagging strand. So the leading strand is continuous. It is working in the direction that DNA polymerase wants to work. Lagging strand, technically, DNA polymerase is still working in the direction it wants to work, but it can't do it continuously because it has to wait for helicase to open up a little bit more. The key part here is understanding DNA polymerase does not switch to working 3' to 5'. It's still working 5' to 3', but it is doing it in a manner that is discontinuous. Okay, so this animation is really good. We're going to talk through it. This top one is forming the leading strand. You can see it's all continuous all the way down. Still has to wait a little bit for helicase to open up, but we're not having to start again. So that is the continuous strand. The leading strand is in 5' to 3'. Now the bottom strand here, it's the DNA polymerase will sit on top of the 5' to 3' parental strands to generate a daughter strand that looks like it's in the 3' to 5', but it is still being synthesized in the 5' to 3' direction. I also want you to notice all these little green things. These are RNA primers. So in the leading strand, you just need one RNA primer, whereas in the lagging strand, you're going to need an RNA primer for every single Okazaki fragment. So this is a little bit less efficient. But it works. That's what we do. Okay, so that was DNA polymerase 3, understand the directionality and leading versus lagging strand. Let's talk about some other polymerases. So DNA polymerase 1, this is going to remove the RNA primer. I'm trying to make a DNA strand, so I don't want RNA nucleotides in there. So DNA polymerase 1 removes that RNA primer and fills it in with DNA nucleotides. So polymerase, you probably already guessed, polymer, root, you're making a DNA polymer. Okay. Now we also have another enzyme called ligase. Ligase is going to bond those Okazaki fragments together. So you can see here in the animation, which I love, that backbone is broken, and I need to basically restore it, which means that I need to put a covalent bond in there. Well, if I'm trying to form a bond, that takes energy, so we need ATP to come in. ATP is going to be used to form that bond, and that's pretty helpful. Okay, it's pretty funny. Take a look at it, take a pause, laugh at it. Let's keep going. Okay, so let's talk a little bit about when things can go a little wrong. So DNA polymerase 3 is pretty accurate. Only an error in about 1 in 100,000 to 1 in a million base pairs. But that's not great considering our whole genome is about 6.4 billion bases, 3.2 billion base pairs. That's not great. I don't want that many errors every time that I replicate my DNA. And so we need to have another enzyme. This one is DNA polymerase 2 that proofreads the DNA. So in case you're keeping track, DNA polymerase 3 is going to be the major player. It adds the vast majority of the nucleotides. DNA polymerase 1 removes that RNA primer and replaces with DNA nucleotides. And then DNA polymerase 2 is going to proofread that DNA code and then check for errors. Okay, so they play a role in something called nucleotide excision repair. Sounds like a tough word, but it actually makes a lot of sense. To excise means to cut out. So to cut out a nucleotide and repair it. So this nuclease, remember nucleases are enzymes that cut DNA, and they are going to cut out the incorrect pairing of nucleotides, and then DNA polymerase is going to fill it in with the correct one. And now our error rate is significantly reduced. Only 1 in 100 million to 1 in a billion base pairs. So way, 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 way less error prone. By the way, lagging strand creates significantly more mistakes. And so this is going to be used a little bit more in the lagging strand in case you're curious. Okay, if you're curious on how this works, you can imagine we have a mismatched base pair. The nucleotide, the uracil is going to be detected and removed. Then that leaves a hole in the backbone that's going to get replaced by a DNA polymerase. Now, what happens when this goes really wrong? Well, there you can have a disorder called xeroderma pigmentosum. This is a genetic disorder, so you're not likely to get it just sitting around, but it's a genetic disorder. And what you need to understand here is UV from the sun can actually generate thymine-thymine dimers. So this is not a mismatch pair. This is a thymine-thymine dimer. So these are adjacent thymines that end up creating a dimer meaning 2. So this isn't supposed to happen, as you can imagine. That doesn't look right. Those are supposed to be base paired with a different DNA strand. Now, typically we can remove this. This is typical of, this happens to us all the time, but then you have DNA repair enzymes that are supposed to come in and fix this. But some people don't. Some people do not have functional repair enzymes, and then you get this disorder. And it's quite sad because it means that they can't go out in the sunlight. They have significantly increased risk of skin cancers and cataracts. Their hands age very, very quickly. It's very, very devastating. So just letting you know, your enzymes are doing a whole lot of work for you. Okay, so in case you were curious on how that works, your thymine-thymine dimers, they kind of create this bump that can be detected. It should be cut out through nucleotide excision, and then it's repaired if you have the correct enzyme. But if this is broken, it doesn't work anymore. Okay, that is DNA replication. It is a lot. I know this is a long video. Believe me, I wish I didn't have to talk this long, but I want you to recap your entire set of notes. I have those words in parentheses for a reason, so those are terms that you should know before you take any sort of exam. I hope you're having a good time studying, and I'll see you soon. Bye.